Welcome to the lecture series on vitamins and nutrition. Today we are going to start with an important vitamin, that's biotin. So we are going to look at one more fat soluble vitamin. So today's content would be, what is this biotin, its historic origin of biotin, and then the sources of biotin, and then the metabolic role of biotin, how biotin is useful. And we would see whether we can finish with that. Otherwise, we will continue with one more session on the deficiency diseases of biotin. Now, let's move on to this particular vitamin, biotin. So the name looks very interesting, right? You do have other important vitamins. We have not seen folic acid, we have not seen pyridoxine, niacin, and pantothenic acid. Many more are there. But I started with biotin because I thought I can explain biotin. Role of biotin, although it looks like a kind of vitamin, we don't have much deficiency as such for biotin because biotin is well supplied by many dietary sources. But outside the vitamin world, biotin has excellent application. We are going to see how biotin would be utilized and various interactions of biotin with other biomolecules can also be seen in the context of vitamin. Now, if you look at the historic background of biotin, biotin is called vitamin H. So you may wonder, what is this H? So if you look into the etymology, it gets from the German word Haar and Haut, which means hair and skin. So since it's an important vitamin connected with development of, I would say, maintenance of hair and skin, and most of the toiletry preparations and cosmetics also include biotin as an important component for the cosmetic industry. So biotin is more used in that line because as such we don't have much dietary deficiencies, although as I have been telling in a routine manner in many previous sessions that there would be a phase, what we call it as persistent deprivation of a particular vitamin because of a dietary habit, or it could be circumstantial. So in such cases, we always show or we can always detect symptoms of biotin. But water-soluble vitamins can always be given as a supplement because excess of water-soluble vitamins are not going to cause that harm as you would expect for a fat-soluble vitamin. So in that way, biotin, being vitamin H, is being profusely used in cosmetic industry, especially in shampoos and other preparations which claim that they would enhance the growth of hair and would improve the texture of skin. So you see there, where vitamin biotin is useful. Biotin as such, if you look, the biochemical role of biotin has many facets and they are utilized as an important component in the carbohydrate, amino acid and other essential macronutrient metabolic process in our body. Okay, so we are going to look at the historic note of biotin. We have close to almost 5 scientists who have contributed in the discovery and characterization of biotin, and you might be familiar with Paul Gayagi. He was also the person we saw earlier when we studied about riboflavin, and his contribution was more into consolidation of the findings of different groups, because always in the history of science you find different groups independently work and they do discovery. Sometimes each one might take a different path and they may not be like really aware of where they head on, and the results what they get could be sometimes synonymous, and that was the case for biotin. Way back in the First World War, when there was a huge demand for nutritive rich foods used by soldiers in the trenches, and there would be mass movement of soldiers, so there was always a demand by the government to provide food because they have to be away from the place for a longer period of time. So there was more of nutrition-based research happening. And here you find in record, W.G. Batman actually had done an experiment by giving a diet rich in egg whites. Yeah, it's true. Egg white is an excellent source of protein. In fact, for people who suffer from, I would say, lipid syndromes and who have been restricted for the intake of cholesterol, are recommended to take egg whites. It's true, egg whites are really good. So what they have done here would be, Bateman's experiment was to look for the symptoms when you feed animals and mammals, especially dogs, cats, only with egg white for a long period of time. You find that they develop certain toxic symptoms. Okay, so this experiment continued. The early phase of nutrition research and vitamin research especially, it was more into a kind of a feeding experiments, as we have seen earlier with all the fat-soluble vitamins and thiamine and riboflavin. The trend continued. And here you find Margaret Avril Boas doing one more set of symptoms, and now the focus would be on rat model, the rodent model, where the animal is supposed to have developed symptoms of dermatitis, alopecia, and muscular coordination problems. So dermatitis is like it starts losing all the fur, and inflammation sets in the skin, and yeah, this is the classical alopecia, when it starts losing the fur in patches, and there would be lack of coordination. See, one more thing we should always consider from the early experiments, although they had used rodent models as a replica for mammalian output, some of the experiments may not sometimes be completely translated, as we have seen in the earlier session, as was the case with vitamin E, where you find there is, I would say, abortion kind of symptoms. But here you find, in this case, the symptoms were almost, I would say, resembling the symptoms what you might observe in human beings when they are deprived of biotin and biotin family of, I would say, nutrients for a very long period of time. So you find this particular symptom of dermatitis, alopecia, and loss of muscle coordination as something what you call it as egg white injury. Okay, so they just characterized it as egg white injury. You may ask whether people would suffer from egg white injury. Yeah, they might suffer from egg white injury in case if they take only egg white and you deprive them of all essential nutrients from vegetable and other sources. So you see here, egg white injury has been bothering, and you find our Paul Ghiaghi confirmed that there is a protecting factor which he called vitamin H from the egg white injury. In other words, the idea was trying to shape up that the yolk had a particular protein and the egg white was deprived of that particular protein. So when you feed them only from egg white, the essential nutrient which was there in the yolk was not available, and people, or I mean in this case animals, started showing symptoms. Now you find here in 1936, as these experiments were going on, you find this group, basically the Kolg and Tonis, had also have looked up the egg yolk as a source to rectify the symptom of egg white injury. So they call it like Bios Eigelb. So Bios Eigelb, basically the life material or vitamin material from egg yolk. So you also find parallelly West and Wilson. They're also trying to isolate a different nutrient, as they called it or they claimed, from yeast and rhizobium. So one group were trying to isolate a vitamin from egg yolk, and the other group were trying to isolate an vitamin from yeast. So the group which isolated it from yeast called it as coenzyme R from rhizobium. So they call it like coenzyme R, whereas the egg yolk group called it as Bios Eigelb. So Georg confirmed both the investigations. He found if you could supplement either biosaucygalp or coenzyme R, you could reverse the egg white injury, and he named it as biotin. So this is a very interesting history. So you see, the one which we use right now as biotin, well characterized structure known, had been discovered over a period of more than a decade. Like the work started way back in 1916, and the final consolidation came only after the Second World War, like in 1940. So you see, the early days, people had done lots of research by vitamin deprivation experiments to ascertain the role of individual vitamins. And now with the advent of crystallography and modern day NMR, we are able to visualize the structure of biotin. And this is the structure we saw, a very interesting structure. We are going to see more of the chemistry aspect of biotin. But before we move on with that, it would be good to look at the dietary sources of biotin. It is true, egg yolk is a very nice source of biotin. You also find biotin in oats, spinach, green vegetables, wheat, germ, white mushroom. We have other sources as well. You have vegetables providing also source for biotin. See, this source of egg yolk is the primary source, or you say for vegans, they can always look for other sources. Honey is also a source of biotin. You have peanuts, molasses, unprocessed sugar. You have the jaggery, the traditional unprocessed sugar, and palm jaggery. All they are rich in biotin. And here you find a whole lot of cereals are also an excellent source of biotin. In general, if you're not like really sure which dietary component I need to take for water soluble vitamins, you will keep seeing this picture again and again till we finish with this lecture series, because most of the vitamins and minerals that we are looking at can be obtained from our normal diet. So that's one of the reasons why we insist on balanced diet, a diet which has a mixture of cereals, a mixture of vegetables, and also an exotic mixture of honey and other stuff. You get all the required nutrients and minerals. Whereas if people have to stick on with some specific diet, that's where all the problem lies. So they, over a period, they start losing the essential vitamins and minerals. So that's one of the reasons either it should be a cyclic diet or a balanced diet where we are being catered with wide variety of minerals. Now, if you look into the structure of biotin, it's a very interesting structure. You have 3 components present over here. So if you have to technically define, or you have to describe the chemical structure of biotin, you have to say it's a heterocyclic ring and a monocarboxylic acid. Okay, so this is the monocarboxylic acid. So what is this acid? It's a valeric acid. See, the name is very interesting because valeric acid means you have almost 5 carbon atom. The amino acid valine is also a derivative of valeric acid. But in case with valine, you have a amino group in the primary position, in the region. So this very word valine basically comes from the flower valine, because it has that kind of a flavor which provides valor. Okay, so that's how the word comes. So valeric acid is 5-carboxylic acid, and this is the key acid which is associated with the heterocyclic ring. So when you say the heterocyclic ring, again you have 2 components. The thiophene ring is there with the sulfur moiety heterocyclic atom over here, and then the urido ring where you have a imidazole kind of a moiety. So you see there, it's a fused appearance. You have a urido ring. So urido ring is, can you see over there? If you carefully look, you can see the skeleton of urea over there. That's why it's called urido. You see NH carboxylic group and then the NH resembling the urea. So this is fundamentally the imidazole part. And then you have the thiophene part. These both are fused with each other, and then they are connected with this carboxylic acid. the valeric acid side chain. So you see this small vitamin has three important components: valeric acid side chain, thiophene moiety, and then the urido moiety. So you see there biocytin, which is basically, I would say, a coenzyme. Here you find the biotin incorporated. So you see there lysine residue being coupled with the carboxybiotin. Okay, so the carboxy part consists of the biotin. So biocytin is a coenzyme that has biotin covalently linked to the lysine residue. So you see there the carboxybiotin. So if you look at the structure, you find the carboxybiotin, which is basically the biocytin. In other words, how biotin gets into the functional form inside our system is in this manner, the carboxybiotin. Now we are going to look at the biochemical functions of biotin. So biotin is very important for carboxylase enzymes, and it acts like a coenzyme for some of the enzymes. So all these carboxylases we have picked up. So for any carboxylase, you find biotin as a coenzyme. So what is a carboxylase? Carboxylase is, you're going to add a carboxy moiety, so you call it a carboxylase. So we are going to look at one example over the other. So you have these four enzymes: pyruvate carboxylase, acyl-CoA carboxylase, propionyl carboxylase, beta-methyl crotonyl-CoA carboxylase. Some of the key enzymes in our normal biochemical pathways which utilize biotin as a major coenzyme. All the carboxylation reactions are dependent on biotin. Sometimes you also have biotin independent carboxylation reactions. So it's not like you have mandatory that all carboxylation reactions have to have biotin. There are some exceptions to the rule. Say for example, carbamoyl phosphate. In case with urea cycle, they do not use biotin. Similarly, for purine synthesis, when you want to incorporate the carbonyl moiety, we don't use biotin. But for these enzymes which are adapted to use biotin, they are extremely useful as a carrier molecule. Now, if we are going to quickly recap the role of biotin, biotin plays a very important role in immune system, in the growth of hair, and in the proper pigmentation connected with skin and hair, and also in the nervous system. You're going to see one by one. Now let's see this particular enzyme, pyruvate carboxylase. So where do you find this enzyme? So pyruvate carboxylase is there where you have to convert pyruvate into oxaloacetate. So pyruvate is a tricarboxylic, I would say, keto acid that needs to add one carboxy moiety over there. So you see there, this part of it, the methyl group, you are adding a carboxy moiety, and this reaction involves energy, ATP. This particular reaction is one of the key reactions in the pyruvate carboxylase enzyme, playing a role in citric acid cycle. So you see there, the pyruvate needs to be converted into oxaloacetate. So this is a classic carboxylase reaction. So you see there, biotin is essential. So you see that the biotin enables in the transfer of carbonyl group over here, and there is an addition of extra carbon. So you see that previously we had only 3 carbon, and now you find 4 carbon. So this addition of carboxyl group, biotin acts like a carrier. Now you might ask, what is the role of oxaloacetate? So oxaloacetate is the key, I would say, the enzyme catalyzed product, which is the raw material that enters and is useful in variety of metabolites. For example, aspartic acid. Okay, this oxaloacetate, once you add an amino group to it, you get aspartic acid. So for aspartic acid, you require 4 carbon. Normally, oxaloacetic acid, if you have to look chemically, it's fundamentally a kind of a succinic acid with the keto group. So in case with pyruvic acid, it's basically a propionic acid with a keto group, and oxaloacetate is fundamentally a succinic acid with a keto group. In both the reaction, you find biotin playing a crucial role in the conversion of carboxyl moiety. Now let's see this part where you have oxaloacetate acting as a raw material for producing citric acid. So citric acid, you know, it's a tricarboxylic acid. So in the citric acid cycle, oxaloacetic acid proceeds further via citric acid and completes the reaction to generate ATPs. Okay, it's one of the important pathways for production of energy. And here you find one more enzyme, phosphoenol pyruvate, in gluconeogenesis. So what is gluconeogenesis? Gluconeogenesis is when your body is deprived of glucose, your body tries to make glucose because your original metabolic pathway needs carbohydrate inputs. So carbohydrate needs to be broken down to produce acyl-CoA, and this acyl-CoA, when it is deprived from dietary lack of carbohydrates, a body tries to synthesize glucose from fat and from proteins. So that's what we mean by gluconeogenesis. This all proceeds via oxaloacetate. So oxaloacetate is converted into phosphoenol pyruvate, and thereby this enters the gluconeogenesis. So you see there, so biotin is such an important cofactor, coenzyme for pyruvate carboxylase. So you see, if there is a deprivation of biotin, consider this entire pathway, okay, where oxaloacetic acid being the important raw material as an input in material is not there, and thereby you have problems connected with all three. Although aspartic acid is not essential amino acid, it means what? We are dependent on aspartic acid through our own metabolic process of synthesis. So biotin deprivation can now make us also be deprived of an essential an important amino acid. Then you have problems with citric acid, and also you have problems with gluconeogenesis and other pathways. So in spite of taking a macronutrient, just a deprivation of a micronutrient, biotin, can actually halt the normal metabolic process. So you see how essential they are. Now you find one more role that biotin plays in the fatty acid synthesis. So you see there in the fatty acid synthesis, biotin is a cofactor for acyl-CoA carboxylase. So acyl-CoA is basically an important input raw material which gets converted to malonyl CoA. So for this reaction, we need ATP, and yes, obviously the carbonyl molecule comes from the carbon dioxide, and thereby you have a malonyl CoA. So here for carboxylase, acyl-CoA carboxylase, biotin is a carrier. As in the previous reaction, this reaction is dependent on biotin. So malonyl CoA in turn has a very important role to play. So you see over there the mechanism how this happens is basically the enzyme bound biotin now acts like a carrier for the carbonyl group. Now this carrier carbonyl group, enzyme activated carrier carbonyl group, allows the transfer of carbonyl to the acetyl-CoA. And the moment that happens, you get the malonyl CoA structure. So you see there, malonyl CoA is basically addition of one more carbonyl group in the form of CO2 to the acetyl-CoA. So you see there that thereby you keep on, I would say, adding up. So for every, so what is basically fatty acid? So fatty acid is fundamentally a long chain acids, long chain carbonyl carbon molecules attached to a carboxylic moiety. So acyl-CoA is the building block So you keep on adding one molecule of acyl-CoA every now and then, and thereby you build the full structure of fatty acids. So you see there, the malonyl-CoA conversion forms the key step for the activation process to occur, and the enzyme which does this is acyl-CoA carboxylase, and this needs biotin. So you see there, the fatty acid synthesis is also affected in case with biotin deficiency or lack of biotin availability. Now you see in one more case, leucine metabolism. So leucine is a amino acid, and you find there beta-methyl crotonyl CoA carboxylase. This is one more enzyme where you have a beta-methyl crotonyl CoA which gets converted, and you have the glutaconyl CoA. So you see there the crotonyl derivative getting added with one more carboxyl group. So instead of a crotonyl, you get a glutaconyl CoA. So this is also a reaction eventually proceeds for utilization of leucine. Now we are going to look at one more reaction whereby propionyl CoA carboxylase. Okay, so this is again an enzyme which is dependent on biotin. So you see over there, citric acid cycle and gluconeogenesis use succinyl CoA, and now how succinyl CoA is formed? Here we start with the odd chain fatty acid cholesterol and the methionine, threonine, isoleucine and valine. So this is like the fatty acid stores and the protein source. So if you recollect, in gluconeogenesis you can depend on two other sources for conversion. So all will converge in the production of propionyl CoA. So this propionyl CoA has to be converted into methyl malonyl CoA, and here you have the propionyl CoA carboxylase enzyme utilizing biotin as a source. So biotin dependent propionyl CoA catalyzes in the presence of ATP to form a 4-carbon group. So you should consider wherever you want to add a carbonyl group, an extra carbon to build the chain. Okay, so you need this. So you see that in this step, the last step we find methyl malonyl CoA. Again, there is one more vitamin B12 playing an important role to complete the reaction. So we have seen in this part, probably we can discuss in detail when we are looking at vitamin B12. So I'm going to give a quick summary. Students can take down the question for today's session. So give a note on the role of biotin, the discovery of biotin, mention the structure of biotin and all the what are the components or the functional moiety of the biotin structure. Then you can take down and mention what are the dietary sources of biotin, and mention the mechanism of biotin involved carboxylase reactions. So you can mention any of these reactions, whatever I have mentioned here, all those carboxylase enzymes you can cite, and you can say how biotin is playing a very important role in the normal citric acid cycle and gluconeogenesis pathway. Thank you once again for your kind attention.